All right, good morning, everyone, and welcome to the Invasive Species Center's 2021 Ontario Invasive Species Forum. My name is Kristen Palionis, and I am a Program Development Coordinator at the Invasive Species Center, and I will be your moderator for this session. Before we get started, there are a couple of things I would like to mention. There'll be time for questions at the very end of the session, so if you have a question at any time, please type it into the question box and I will get to this at the end. And then if you're having any technical difficulties at any time, please type them in the question box and we will try to resolve it for you. So this morning's session is titled Invaders of Ontario Forests, and I am pleased to introduce our first speaker, Chris Macquarrie, who will be presenting on invasive forest pest research at Natural Resources Canada's Great Lake Forestry Centre. Chris Macquarrie is a research scientist with the Canadian Forest Service at the Great Lakes Forestry Centre in Sault Ste. Marie, Ontario. He studies the population dynamics and management of invasive and native pests of trees. This presentation will provide a brief summary and update on current invasive pest research at the Great Lakes Forestry Centre, including work on emerald ash borer biological control, hemlock woolly adelgid detection and management, and other important invasive forest pests. So thank you, Chris, for joining us this morning, and I'm going to hand it over to you.
Cool. Which screen can you see?
Right now, it's we see your notes.
Yeah, I need to share a different screen. Oh dear. I used to know how to do this. Hang on a second. Could you offer me to show the screen again and I'll pick the right screen this time? Oh, there we go. Hang on a second. There we go.
Looks great.
There we go. Sorry, folks. You're into this. You still think you'd think I'd know how to do this by now. Okay. Thanks for tuning in today, everyone. As was mentioned, I'm going to give you a brief update on what we're going to be doing on some of the research that goes on at the Great Lakes Forestry Centre on invasive forest pests. And this is, I'll talk a bit about some of my own stuff and then some of the work some of my colleagues are doing. So I will give, there we go. So just a brief outline of the talk today. I'll give you a bit of a quick introduction to the Canadian Forest Service to folks that maybe aren't familiar with us. Talk a little bit about what pests, some of the pests we work on, some of the invasive forest pests that we work on, and then a bit of some highlights from some of my colleagues. So the Canadian Forest Service is part of Natural Resources Canada. We are one of 6 research centers located across the country. From west to east, we have the Pacific Forestry Centre in Victoria, the Northern Forestry Centre in Edmonton, us at the Great Lakes Forestry Centre in the Sioux, the Laurentian Forestry Centre in Quebec City, and then the Atlantic Forestry Centre, which is co-located in Fredericton and Corner Brook. We also have the Canadian Wood Fibre Centre, which is a virtual research institute that has staff in all of our physical research centres. Our headquarters are in Ottawa, and we also have a number of research forests and research sites. A couple of those I've highlighted with the green dots on the map. We do invasive forest pest research at all of these places, and we collaborate on a lot of the work that we do on invasive forest pests with colleagues across the country. So we're not just, so the work that we do is not just centered at the Great Lakes Forestry Centre. This is done in collaboration with colleagues all across Canada and internationally, and then folks at universities as well in this province and in other provinces. So it's a collaborative effort with a lot of different researchers. So the Great Lakes Forestry Centre, like I said, is located in Sault Ste. Marie. That's a view of the lab that as in about 2 weeks we will have been out of for about a year. We're organized into 4 divisions. We have what is called the Integrated Pest Management Division, the Integrative Ecology and Economics Division, and excuse me for a second. And our Forest Ecosystems Division, and then our Partnership Planning and Operations, which keeps the lights on and in collaboration and develops collaborations with some of our partners. Most of our pest management research is focused in the Integrated Pest Management Division and the Integrative Ecology and Economics Division. Most of the sort of on the ground pest management type research is done in the pest management group. Our colleagues in the Ecology and Economics Division spend a lot of time looking at the economic impacts of invasive forest pests, and looking at things like estimating risk of spread and invasion from new pests and existing pests in Canada. So these are kind of the big 4 these days that we spend a lot of our time working on. There's probably not a lot of introduction needed for folks who are interested in invasive species, but the big one at the top is the emerald ash borer, the one that a lot of folks are familiar with. We've got the Asian longhorn beetle there on the right, the very pretty black and white longhorn beetle. The one on the left that looks like the kind of fuzzy stuff on the pine branch, or the hemlock branch there, that's Hemlock woolly adelgid. And in the bottom we have Syrinx noctilio, which is something else that we still work on a little bit. So I'll talk a little bit about what we've been doing on those different pests here as I go through the talk. So talk a bit about some of our research highlights. So talk a bit about myself first, because I'm the one doing the talk. So my sort of long-term goal of my particular research program is looking at, for emerald ash borer, is looking at developing predictive population models for EAB. So can we develop tools for managers and tools for people who assess risk to figure out where emerald ash borer is going to go, what kind of damage it's going to cause, and then tools for helping to manage that, to know when to apply control tactics like biological control, which we'll talk about in a minute, or things like insecticides or other management tactics that might be developed. So developing those tools for managers, and certainly one of the biggest things that we're involved in right now when it comes to emerald ash borer is our biological control program. So just showing here on the screen the 3 parasitoids that we've been releasing in Canada since 2012. This is Ubis agrilli and Spathias gallinae and Tetrasticus plantapenisi. And we do this in collaboration with a whole bunch of folks located across the country, and who've either supported the research or providing us with release sites to establish these parasitoids in a place where they won't get disturbed for a while. And then we work in very close collaboration with our colleagues in the United States who provide us with the parasitoids and have done the lion's share of the work to figure out how to successfully rear these things in captivity and then release them successfully so that they can establish and impact the emerald ash borer. So, like I said, we've been releasing these parasitoids in Canada since 2012, and what this map shows is just the locations of our release sites across Eastern Canada. So you can see we spread all the way from basically Sault Ste. Marie, St. Joseph Island in the east, all the way to New Brunswick, northwestern New Brunswick in the east. Our release pattern has sort of followed the invasion of emerald ash borer. So we started in 2012 way down here in southwestern Ontario, and we've tracked up, excuse me, I'll probably show this on the screen, you can see, down in southwestern Ontario here, and we've released over the years, moving to the east, the last couple of years in Quebec, and then this sort of part of Ontario. So this is where we've been releasing parasitoids, and we release 2 or 3 at each site depending on the year and the site conditions. And we do releases for a couple of years, and then we come back later and look at whether those parasitoids have established and if they're having any impact on the population. That's some of the work we've been doing recently, is looking at establishment and spread of Tetrasticus plantapenisi. So that's one of our parasitoid species. That's kind of the workhorse of the biological control program in Canada. And in over the last 2 years, we've been able to confirm the establishment and dispersal from 4 of our 7 sites. We did this work in 2019, And obviously planned to expand on it last year, but that didn't happen. And so we ran a trapping network at distances from these initial release sites, and we were able to confirm that the parasitoids had managed to spread out into the environment. This is just a picture on the right showing the pan traps that we were using to assess dispersal of the parasitoids, and we're still in the process of going through these samples from 2019 to see how many sites actually had those parasitoids. And then sort of independently, we've also been able to confirm dispersal of some of the parasitoids from our site up near Sault Ste. Marie. We've actually been able to find them right in the Sault. So they've been out there establishing for a couple of years and moving across the landscape, which is we think is good news. More recently, we've been moving into work on Hemlock woolly adelgid. This is a new, somewhat a new invasive in Canada. It's well established in southern Nova Scotia, and there's a lot of work being done out of our lab in New Brunswick, looking at management tactics and the potential for doing things like biological control and then mitigating the risk to other places. In Ontario, we have less of an established population, so it was found in the Niagara Gorge a couple years ago and in and around Toronto, but it was eradicated. But then about 2 years ago, it was found at a site near Waynflete, Ontario, and it's a well-established population. So we're last sort of year and a bit, we've been sort of gearing up to do more work on this bug, and I think you heard about some of this work and some of what's been going on in a session earlier in the week. So some of the things we're looking at is sort of optimizing survey strategies, developing hemlock inventories for Southern Ontario, which is a kind of a gap we want to try and close, looking at different chemical control tactics for HWA, then looking at, and then also we also want to look at, what's the phenology in Ontario, what's the range of it in Ontario, has it spread other places, and that's work we're doing collaboration with the Canadian Food Inspection Agency. And then the last thing we want to do is look at what are our resident natural enemies for HWA in Ontario. So what might eat that bug if it was just to establish here more and start to spread. And that's sort of preparatory work for thinking about doing biological control of HWA. There's a well-established biological control program in the US. And at some point, that's a tactic that would be of use in Canada. But we have to do the work ahead of time to see if what natural enemies are here and what work we'd have to do to assess risk of introducing those biological control agents into Eastern Canada. So that's a bit about my stuff. I'm not the only researcher at the Great Lakes Forestry Center. We have a team of 8 or 9 people who are in our pest management division, and then another 2 or 3 other scientists in the economics division that work on invasive species. And I'm just going to talk a little bit about 2 of my colleagues that are actively engaged in sort of research on invasive species. And one of my colleagues is Dr. Jeremy Allison. He's our resident chemical ecologist, and a lot of the work that Jeremy does looks at optimizing the effectiveness of insect traps. And this is tools we use to monitor for the detection of new invasive species and assessing the dispersal of species that are already resident in Canada. So a lot of that's the integration between how the insect responds to its environment and to things like baits and lures, and how traps are able to be more effective at capturing these insects so that we know that they're in the environment. So a lot of stuff that Jeremy has done is looking at how and why insect traps differ in performance. And so we sort of have a bunch of different classical designs here for insect traps. These are the classic panel traps and the lingering funnel traps that are used in a lot of different forest insect surveys. Oh dear, what's going on? Okay, it decided to go on its own. This is what happens when you get slides from other people. So one of the things that Jeremy's looked at is looking at trap design factors affecting the performance of those traps. So on the left, this is some results from an experiment that was done up here in Northern Ontario, looking at canopy closure, so open versus closed canopy, just work done in a cut block, and then looking at different types of panel traps. So this is the design on the bottom, looking at, when do you catch more oh dear. I don't like slides that advance on their own. And this is looking at the response of Monocamus, which is sort of a stand-in species for some of for longhorn beetles and for these other insects that we're interested in trapping, these invasive insects that we're interested in trapping around things like ports. So looking at what's the best trap to use for these kinds of programs. Then the work on the right is looking at how when you're using a baited trap, so an insect trap that has some kind of chemical bait in it to attract an insect, how the plume of that pheromone or the plume of that bait is emitted from the trap, and that can affect how the insect responds to it, so its orientation, behavior. And this is some work that was done by Mark Bauer, who was a postdoc from South Africa, working at the Great Lakes Forestry Center for a couple of years, and is now in private industry in South Africa, looking at how sort of a stand-in plume, so a carbon dioxide trace, was emitted from this panel trap, and the idea of, how would you hang this trap in a forest stand to see and how would the insect respond to that, depending on which direction it was coming from. So it all goes back to optimizing those traps. Some other work that Jeremy's done is looking at the impacts of Cyrex noctilio in North America. So Cyrex is a pest of pines that's been widely distributed across the world, really. It's especially in the global south. One of the interesting things about Cyrex is that it is not, at least in Canada, has not become sort of the insect pest that we thought it might have, but it certainly is in the global south, places in like South Africa and South America. And so some of the work Jeremy's done with collaborators in South Africa at FABI is looking at what's the difference in emergence and brood production when you artificially protect infested logs from predation by natural enemies. And so that's kind of the picture you see there on the left. So using cages and various other manipulations to either let enemies or not let enemies attack these logs. And what you see is that when you have when you look at brood production in these experimental logs, is that you get a very big difference in how many insects emerge, depending on whether you're looking at this experiment in North America, which is the top panel, or South Africa in the bottom panel. So since you see very little emergence in North America, what that means is there's lots of natural enemies that are attacking these insects and preventing them from developing. But that natural enemy community doesn't probably exist in South Africa, and so that's why you have it. That's probably part of the reason it's such a big problem in these other places. So this is an example of this collaborative research we do with folks in other parts of the world and other parts of the country to look at the effect of invasive pests, because certainly things that are invasive here are invasive pests everywhere, and we're always looking to learn from other places. So next thing I'll do is I'll talk about some work by my colleague Amanda Rowe, who's an insect molecular geneticist here at the Great Lakes Forestry Center, and she leads a lot of our work on Asian longhorn beetle, which is the big beast you see there on the right. Certainly a big pest that was found in Toronto a number of years ago and eradicated, and then found again, and then eradicated again. And some of the work that Amanda's been doing is looking at tracing the source of invasions with genomics. So trying to figure out where have these populations, so Asian longhorn beetle originates in China, and has been introduced into a number of places around the world over the years. And some of the work she's been doing is looking at the genomic signature of these populations and trying to figure out, when we get an invasion, can we say where it came from, right? And once we can say where it came from, we can look at what mitigation strategies do we need to use to prevent those invasions from happening again. And then some of the other work that Amanda has been doing is looking at cold tolerance of invasive species. So as folks knows, we've just gone through winter. Winter is a big thing in Canada. It's a big constraint on the success of our native insect species and our invasive pests. So if you can understand how invasive insects cope with low temperatures, you can start to make predictions about how well they'll be able to survive under our climate and under a changing climate and in different parts of Canada. So this is looking at things like how insects enter diapause, what physiological features they have that can allow them to survive our climate, and then whether these features of their physiology can be subject to selection. So can they be changed over time? So and then that all goes back to sort of predicting that invasion risk that for invasive insects coming into Canada, so we can start to have an idea of what insects might be a problem and how much of a problem they might be. So that's a very quick overview of the kind of work that we do at the Great Lakes Forestry Center, and certainly this is just a snapshot of some of the stuff that just the three of us are doing. There's a large group of researchers that touch on a whole bunch of different aspects of invasive species, and sort of always interested in talking to folks about what research questions they might have, or what pest management problems they might have, and in terms of being able to answer those questions, or figure out what we can do to answer those questions. So with that, I'll leave it. The last slide I'll pop up is just the contact information for the research that I showed. For the folks that have done the research that I showed on the past couple of slides, and certainly I'd encourage you to reach out to any one of us, or to any one of my colleagues at the Great Lakes Forestry Center if you're interested in knowing more about what we do there. So thank you, and with that, I'll stop sharing my screen.
Thank you, Chris. That was a great presentation, and it seems like you and your team are very busy with all the work that you're doing. So that's great to hear. Thank you. All right, so our next presenter is Dr. Sharon Reed, who will be talking about emerging forest pathogens, including oak wilt and beech leaf disease. Dr. Sharon Reed has been working as a forest health research scientist for the Ministry of Natural Resources and Forestry for nearly 3 years. The lab's current efforts are focused on detection, survey, and management of invasive species. Topics include oak wilt, beech leaf disease, and phytophthora. Oak wilt and beech leaf disease are two new emerging threats to Ontario's forests. This presentation will discuss why these diseases are a threat and the signs and symptoms of the diseases. In addition, the audience will learn about current research aimed at improving detection, identifying risks, and developing management. So thank you, Sharon, for joining us this morning, and I'm going to pass over the controls to you.
Okay. Okay. All right, can you see the full screen now?
Yeah, it looks great.
Okay, great. Okay, so I'm going to start out by talking about oak wilt. So this is a disease that was first described in the 1940s in Wisconsin, but at that time it was already recognized to have been present much earlier. And indeed, when they started to do more research in other states, they found it was already present in Ohio and West Virginia and Missouri. Since that time, the natural spread has been fairly slow, except for on this diagram, you can see that there are these red detections outside of the larger range, and those are thought to be due to the human movement of infected wood. Oak wilt has not been detected in Canada, but we do have reasons to be concerned. You may have heard that in 2016, oak wilt was detected on Bell Island, which is 500 meters from Windsor. It's been present in Michigan for decades. There's a map here on the right-hand side of Michigan. All the red dots are the confirmed detections, orange are reported, and blue are treated oak wilt pockets. So you can see that they have quite a bit of it. There is probably more. This map is based on volunteers and submitting data. It's also been it's being found more frequently in New York as well. There are some detections. You can see these blue dots on the left-hand side map. They are just a few counties away from the Niagara region. And I'll be talking a little bit more about this later on in the research section. But we were doing a study along the Michigan-Ontario border to develop a DNA-based tool for detecting oak wilt, and we were able to find DNA from the beetles on beetles that we trapped at the border, no oak wilt was found. No oak wilt infected trees have been found in Canada. Those beetles may have come from Michigan and been windblown here. but the detection of the DNA definitely shows that we have a reason to be concerned. Okay, so oak wilt is caused by an invasive fungal pathogen. It causes a wilt because it colonizes and plugs the water conducting tissues of the trees. In this photo is a picture of the fungal mat that the fungus produces on dying trees or freshly killed trees, so in the fall and the spring. The fungal mat is actually a pressure pad. It actually pops the bark off of the tree, creating a crack that allows insects to crawl down into it. And the fungus has a sweet smell to it that's attractive to insects. The most common vector of the oak wilt fungus are knitted-dooley beetles. They're also called picnic beetles or sap beetles. They're attracted to that sweet smell, and they're attracted to fruits and other things that smell sweet as well. You can find them in your gardens as a pest often. They crawl, feed, and lay eggs on the fungus. When they become contaminated with the fungus, they leave the fungus and the diseased tree, and they are also attracted to wounds on healthy trees, and that's how the transmission of the disease occurs. There are some other insects that might play a role in oak wilt in the southern US, but the sap beetles have been found to be the most consistent vector in northern areas of the United States. All trees in North America are susceptible to oak wilt infection, but they differ in their susceptibility. Trees in the red oak group, like a black oak or a red oak, are the most susceptible. They can die within a matter of weeks of being infected by the fungus. Trees in the white oak group are less susceptible because they produce tyloses that are able to wall off the infections. And there have been some reports of chestnuts, Chinese chestnut plantations having infections, but that appears to be rare. So the symptoms of oak wilt, one of the sure signs, these wilting red oaks have early leaf drop. In July, usually when we just have our normal drier period, in the summertime, those trees are already suffering because the water conducting system is plugged up, and they start dropping their leaves, usually from the top of the crown downwards. You also can get this characteristic dieback in the leaves. They dive back from the tips to the midrib. And in the bottom right photo, we have the blue staining of the wood that can be seen when you scrape the bark off. And there are differences between the red oaks and the white oaks. You know, white oaks, because they can wall off the infection sometimes. Sometimes you'll see branch dieback in those trees, although oak wilt definitely can kill trees in the oak wilt group as well. There are two major pathways for oak wilt. The above ground pathway, we've discussed a little bit with the insect vector. So the bottom of this diagram is your recently killed oak wilt tree that produces the pressure pads in the spring or the fall. It pushes the bark off, the beetles crawl down in it, they become contaminated, then they infect a healthy tree that's been recently wounded, a wound that goes down to exposes the wood of the tree. And then you see symptom development, and you're back at your dying tree producing the new oak wilt mats. But there's also a below ground transmission pathway, and that's where treatment of oak wilt becomes time consuming and expensive. So when a tree dies, red oak tree dies, the fungus goes down into the root system. And the root system stays alive for multiple years when the overstory has already died. It can pass through grafts between the diseased and the healthy red oaks, or the same pathway for white oak trees in the white oak group. And so what you end up with, look at this top photo with this aerial survey that's been done. You end up with these disease pockets. They've lightened 6 areas on the photo just to accentuate so you can see better these pockets where you have dead trees in the middle, and on the outside edge are trees that are infected and dying. Okay. And the way that to stop the spread of those pockets is actually to break up all the root grafts first. And you can do that with these huge pieces of equipment, these vibratory plows. Also can be done with bulldozers if it's a rocky situation. Chemical treatments have been used to break root grafts in situations where you just can't use these large pieces of equipment. And then you have to remove all of the dead trees and dying trees, potentially infected trees above ground so that they don't produce more fungal pressure pets. And then you have to return on a regular basis to make sure that all the infected trees were truly removed from that area. There are no chemical cures for oak wilt infected trees. There is a fungicide that can be injected into trees in the US that's available. However, nothing stops oak wilt's infection once it's started in a tree. Those injections are preventative. So for some of the research that we are doing, I already mentioned briefly that we are working on with Natural Resources Canada and CFIA, as well as the Quebec ministries, to develop a highly sensitive test to detect DNA of the oak wilt fungus in various situations. This will help us understand where our highest risks are, so that way we can find ways to mitigate those risks. We did have during our pilot, our test project, the detection of DNA at the border for Ontario. And I would stress again that this is a good news story, that this detection means that the test works. But we have not found any oak wilt infected trees in Canada, and those beetles may have originated from Michigan. Right, and the next work that we've been contributing to Ontario Ministry of Natural Resources, working with Natural Resources Canada scientists, was to understand the risks and cost of oak wilt establishments. And the paper was published in 2020. I think John Pedler may have just given a presentation for the invasive species seminar series just last week or 2 weeks ago. So one of the major findings was that the climate of southwestern Ontario is already suitable, and that by 2050, most of Canada will have a climate suitable for oak wilt. So again, we need to be prepared for the possibility of oak wilt entering Canada. And you can also go to this paper to look at more of the costs associated with removal of trees. Our lab for the Ministry of Natural Resources and Forestry has been leading a project along with Natural Resources Canada, Manitoba Sustainability and Development, and OMAFRA, looking to identify the potential vectors of oak wilt in Canada. So we should say that there are many different species of sap beetles in North America. And in the States, they have found that the beetle species that are the most important vectors varies by region. So we've been wounding red oaks and bur oaks throughout Canada to better understand which insects may be our important vectors. We have found 12 species associated with red oak wounds and 1 species associated with bur oak wounds. And we have the 2 primary vectors. We found that the 2 primary vectors from the Midwestern US, from Wisconsin and Minnesota, are widely spread in Canada as well. So those are definitely suspects for us. And last but not least, part of that project is also following insect flight from March to November in Southern Ontario, Northern Ontario, Manitoba, and New Brunswick, and collecting air temperature data along with that flight to build degree day models to estimate the start of the flight period. This will allow us to determine when our risk of oak wilt infection is occurring and provide guidelines for when to avoid wounding oaks to limit the risk of oak wilt infections occurring. So the next disease I wanted to talk to you about is beech leaf disease. You can see these stripes on these beech leaves in this photo. This is a disease that's present in Ontario. Beech leaf disease has already been detected in 7 states in the United States. It was first detected in Ohio in 2012. We now know in Ontario that it is spread along the shores of Lake Erie as well as in Toronto. The first detection was made by one of our Ontario Ministry of Natural Resources and Forestry's forest health technicians in Elgin and Northland County in 2017. So it's taken since 2012 to understand what is the causal agent for beech leaf disease, and one of the reasons it's taken so long is because this one is rather unusual. We found a nematode is involved in the disease, and so it looks like there's a paper that was published in March of 2020 with Carleton et al. We worked with the labs in the US to identify that run disease trials that showed that when you spray the buds of beech trees with a solution containing this nematode Lidolingus crenate subspecies McCanny, the leaves that come out of those buds in the springtime are infected with the disease. So then we followed up with another paper, which I did include the link this time on this paper that you go to read more about if you're interested. It shows the distribution where we found the nematode, as well as the types of materials. So we did find that it's present in the beech leaves and buds, and we found that it was able to overwinter in beech buds, as well as the leaves, beech leaves that had fallen to the ground in the fall. So it's not just American beech that's affected by beech leaf disease. It's also was found on European beech hedge in Toronto. It's been found on European beeches in other states, in the States as well. And then there's an arboretum in Ohio where they also found that the oriental beech and the angler's beech had become infected with the disease. So it does infect a large number, and that makes this an issue where there's concern that beech leaf disease could spread from one continent to another. So the symptoms are easiest to see, especially when it's a low level infection. When you're looking up at the leaves and the light is shining through them, you can see these dark bands that are forming between the leaf veins. Those are the feeding sites of the nematode. And the nematodes don't like to cross these vein tissues. So you get these stripes in between the leaf veins from that. This top left photo is a normal leaf. This is what those stripes can look like in just after the leaf out occurs. So kind of a reddish color. Later in the season, you get these darker bands, the bottom right photo. Then after a tree has been infested or infected for several years, you start to get these leaves that more than 2/3 of the leaf is infected. The tissue has become really coarse and leathery. You get this yellow coloration as well. And you can get curling of the leaves and tears in these leaves that are on trees that are infected for a longer period of time. And just because I want everybody to go look for beech leaf disease, this is what it looks like in the fall as well. Rebecca Lidster, our forest health tech, sent these photos up to me in the fall, and I just thought they were really interesting, where the bands, they're retaining their color in the fall, whereas the rest of the leaf has started to take on its yellow color. So you can see here the green bands, black bands, and the yellow coloration of the leaf. So it's really noticeable at that time of year as well. The other symptoms, we do see that there's early leaf drop in the trees that have been infected for several years in a row, and we have a bud abortion that occurs as well. And the infections, mortality in the smaller trees, the saplings, occurs within a couple of years, up to 5 years. Overstory trees are infected as well. And you can see this picture is from Ohio, where the overstory has been severely affected by the beech leaf disease. So research, so we had some questions, first off, is this an invasive species? How similar are all these populations to each other? If this was a native species, you'd expect there to be some small genetic differences in different populations. We found, working with Katrin Fitza and from the University of Pretoria and her advisor Bernard, or her colleague Bernard Slippers, and Jeremy Allison from Natural Resources Canada, we found that the populations of nematodes in Ontario, they're all identical at the different gene regions that we looked at. And that they're also very similar to the nematodes from the United States, suggesting that we had a common source for this nematode. Right now, we're working with David Burke and Adam Hoke from Hodum Arboretum in Ohio to develop a better detection tool, quantitative PCR that will help both with research and detection to know if nematode is present, because we did find that you can find small numbers of the nematodes in tissue that does not show symptoms. So we want to be able to track where the nematode is, since it can be present even if there aren't symptoms. This qPCR will be highly sensitive, and we'll be able to use it directly on the leaf material. It'll be a lot faster than extracting nematodes and counting them. In the final area of research that we're working on for beech leaf disease, we are working with partners in the US to determine the impact of beech leaf disease on forests. We have 34 plots established in southern Ontario along Lake Erie. And this study is designed to look at the impacts of both beech leaf disease and beech bark disease. We're quite concerned because beech bark disease has been establishing itself in Ontario, and it mainly kills the larger trees that have a diameter greater than 10 centimeters DBH. And whereas with beech leaf disease, we see that it's most severe on saplings, so the smaller trees. So we need to better understand what is going to be the interaction of having these two diseases present in our forests. Right, so let's leave you with the thank you slide. There's so many people that have helped with these different projects, and we really appreciate that. Great, thank you.
Thank you, Sharon. That was great. It looks like there's lots of interesting research going on with beech leaf disease and oak wilts. So thank you. All right. Okay, so I would now like to introduce Dr. Catherine Cullingham, who will be presenting on mountain pine beetle and its potential for eastern spread. Dr. Catherine Cullingham is an assistant professor in the Department of Biology at Carleton University. She received her Bachelor of Science from the University of Guelph, her PhD at Trent University, and completed postdoctoral work at the University of Alberta. Her research uses landscape and population genetics to fill knowledge gaps and develop tools that can be applied to issues in forestry and wildlife management. She has been working on the mountain pine beetle system for over 10 years and has contributed to confirming post-expansion to jack pine, defining the spatial complexity of the lodgepole jack pine hybrid zone, and identifying genetic markers potentially associated with mountain pine beetle resilience. Mountain pine beetle is a bark beetle that infests a number of different pine hosts in Western North America. Due to anthropogenic factors, this species has expanded its range into the Western boreal forest. This talk will cover some of the interesting aspects of mountain pine beetle biology, as well as the future spread risk potential for Ontario. So thank you, Catherine, for joining us this morning. I'm going to hand it over to you.
Hopefully everything's showing up okay there.
Looks great.
Perfect. First off, I'd like to thank ISC for inviting me to talk and for all of you today for listening. I'm going to go through a little bit about the mountain pine beetle biology, a little bit about its potential for eastern spread and some of the implications of that spread. I'm not an entomologist, so it will be very high level information. So to start off, mountain pine beetle, it's a bark beetle that infests a number of pine hosts, and this includes lodgepole pine, ponderosa pine, sugar pine, western white pine. And it has a cyclical, life cycle. It has a circular life cycle. So the adult beetles emerge from the tree in the summertime. They fly to find a suitable host. Once they find a suitable host, they bore into the tree under the bark and build galleries. In these galleries is where they lay their larvae. Over the wintertime, the larvae feed off the phloem of the tree and then emerge again in the summertime as adults and find a new host. They have some helpers along the way. So the mountain pine beetle has these specialized structures called mycangia. You can see them in the microscope image on the far right. And these mycangia carry different fungal species. And so these are called blue staining fungi. Some of the usual suspects are Grossmannia clavigeria and Leptographium longiclavatum. And they result in staining the wood blue. So in the central picture of the bolts of lodgepole pine, you can see a blue ring around the outside of them, and this is caused by the fungus. So the fungus benefits from the beetle in that it gets a method of dispersal. So it hitches a ride so it can move around the landscape. And it's thought that the beetles benefit from the fungus when they lay their eggs. The larvae use the fungus as part of their nutrition. So they feed on both the fungus and the phloem. And together, the fungus and the larvae end up exhausting the tree and killing it. The distribution of these fungal species varies across the landscape depending on where you are and climate. When we talk about the beetle, it exists in two different phases. So typically it's in an endemic phase. So these beetles are quite small. If you've never seen one, they're about the size of a grain of rice. So when they are in an endemic phase, population numbers are low and they can't attack or infest really healthy trees. So they tend to infest trees that are compromised in some way. So they could have been struck by lightning, or they've experienced high drought stress, or maybe they're just really old. And so when it infests these, lower quality trees, they don't produce as many offspring. So population numbers are low. And when population numbers are low, stand mortality is also low. Now, sometimes when conditions are right, reproduction is increased. So maybe it's because there's been a couple warm winters and there hasn't been as much mortality of larvae. So in this instance, population numbers are and they can infest really healthy trees. And when they infest healthy trees, they can make more babies. And more babies means more trees being killed. So in an epidemic phase, you'll see a lot of stand mortality. You'll see this at kind of the landscape level. And this is a co-evolved system in Western Canada. And so you might be wondering why we're talking about it being an invasive species for Ontario. And so I'm going to show you a bit of a time series of maps. So in this first time series of map, we have BC and Alberta, We have the species approximations for lodgepole pine, jack pine, and the hybrids between them. And we also have mountain pine beetle incidents. So this is a period of 40 years. So this is the mountain pine beetle outbreaks from 1958 to 1998. On the far right of your screen, there's a graph showing kind of the epidemic cycles of the beetle over about a 100-year period. So you can see around in the 1930s, there's a bit of a spike in the annual area affected in BC. Again, in the 1950s, there's a spike. And again, around the 1980s, there's a spike. So you can see there's this endemic epidemic cycle. This is all natural. However, things changed a little bit. So you can see in this 6-year period from 1998 to 2006, we have magnitudes greater outbreak. You can see it both in the map as well as on the graph. And so this outbreak was fueled partly by climate change factors as well as some other anthropogenic factors. And so this has kind of fueled the spread and had, you know, really big impacts for the lumber industry and the, you know, for the country. I mean, BC is one of our biggest, is the biggest lumber industry. So, you know, really big impacts at the landscape, cultural, social, all sorts of ecological impacts. But what also happened during this time was it spread into Alberta. So the Rocky Mountains were considered a biogeoclimatic barrier to mountain pine beetle. And then when this large outbreak happened, the beetles actually were carried over the mountains by wind, and it rained beetles in Alberta. And so it has spread into this novel habitat. And so this is the incidence data up to 2019. So we've now have this beetle that's expanded into central Alberta. And so what's the spread risk for the rest of Canada? So in BC, it's been primarily attacking lodgepole pine forests. In Alberta, lodgepole pine transitions to jack pine of the boreal forest. And in this transition zone, lodgepole and jack pine form a hybrid. So the first question we needed to address was, is jack pine susceptible to mountain pine beetle? If jack pine isn't susceptible to mountain pine beetle, there's really no risk. And so there's been a number of studies that have been done on terpenes. So terpenes, conifers produce terpenes as part of their defense mechanism. So this is how they fend off invaders. And the graph on the right shows a comparison between jack pine and lodgepole pine. So in the top graph, this is limonene. So jack pine produces a lot less limonene than lodgepole pine. And this is bad. This is bad because limonene is considered more resilient to mountain pine beetle. So it's actually effective against mountain pine beetle. So that's bad. In the bottom graph, you'll see that jack pine produces a lot more alpha pinene than lodgepole pine. So you're thinking this might be good. No, this is also bad. So what alpha pinene is, it is used by the mountain pine beetle as a precursor to create pheromones. And so mountain pine beetle use pheromones to communicate with each other. So jack pine actually produces a lot of this chemical that the mountain pine beetle uses. So this suggests that, yeah, jack pine is, you know, is susceptible, and there are some differences between it and lodgepole pine that may make it even more susceptible. And these differences likely exist because lodgepole is an evolved host with mountain pine beetle, and jack pine is a naive host. And then the second thing that the part that I contributed to was actually identifying jack pine on the landscape in a natural stand that had been attacked by mountain pine beetle. So using genetic markers, we were able to identify trees in the hybrid zone that were pure jack pine. And the picture on the right is some of those trees that were successfully attacked. So jack pine susceptible to mountain pine beetle. What is the likely rate of spread? Is this a problem that we need to think about in 5 years, 10 years, maybe 20 years? So how quickly can mountain pine beetle travel through the boreal forest into Ontario? And this really depends on a lot of things. And I wish I could give you a very clear answer, but I can't. So Barry Cook and Alan Carroll did some really lovely modeling. And what they showed was that there are so many unknowns in the system, it's quite difficult to predict the spread. And you know, their overall conclusions were saying that, you know, maybe that forest management just needs to continue exploring ways of coping with these unpredictable disturbances. And some of the things they highlighted were weather. While we can, you know, project a couple days in advance what the weather might be and we might see trends, we don't know exactly what it's going to do from year to year and how that's going to affect spread. And also something really important is response to novel environments. So mountain pine beetle's gone from BC. We've, you know, there's been a great history of research in BC and understanding of the beetle, but now it's somewhere new. So how do we deal with that? So we've got new research directions to take. And so one of the ways I'm looking at this is I'm trying to examine mountain pine beetle spread in the hybrid zone. So in Alberta, we have this, it's a really complex mosaic actually. So what I've done here is I've used genetic ancestry to predict where hybrids and pure species are going to occur. And this is kind of just a zoomed in spot on the map showing you how complex this is. And so we have, you know, pure lodgepole pine in dark blue. We have lodgepole pine like hybrids in light blue. In orange are our pure hybrids, so they're kind of 50-50 lodgepole jack pine. And then in light green, we have our jack pine like hybrids. And then in dark green, we have our pure jack pine. And what I want to do is I want to use these data to actually look at incidents from year to year to say whether or not beetle spread is being fueled by lodgepole pine only, or if there's evidence to suggest that pure jack pine is fueling spread. So the end product of this work will be tell us, can jack pine support epidemics? So this is a bit of a piece in the puzzle, because while it's susceptible, it might not be able to support epidemics for other reasons. You know, there could be other insects that infest jack pine that affect mountain pine beetle's ability to reproduce in greater numbers. Its phloem might not be as nutritious as lodgepole pine. So there might be a whole bunch of different factors that play into whether or not jack pine can support epidemics. So we're trying to use some modeling and information we already have to start to pick away at this question. And then let's say that jack pine can support an epidemic. The next question we want to ask is, are all trees equally susceptible? So here we have a lovely landscape, and we can see in the foreground we have some, you know, bright red attacked trees and then lots of green trees. So is this just a factor of beetle dispersal, or is there some choice and resilience? So choice on the beetle side, resilience on the tree side. And because I do genetics, we're trying to do this using genetic data. And so far what we've come up with is we've used lodgepole pine as our basis. So we already have, you know, attacked and unattacked trees in lodgepole pine. We've identified genetic markers that vary between them. So we've identified certain gene or certain variants that are associated with a tree being attacked by the beetle. And from that, we've kind of created a top list of markers, and this is what they look like on the landscape. So what we're trying to do is use all of this genetic information to create one stand level risk rating based on genetic data. So I've called it a resilience rating. So in BC, we have a lot more resilience, and that's because we have our evolved host. And as we transition from our evolved host to our naive host, we have fewer of these resilient markers. And so this is the kind of research that we're just starting to unravel, but this is what we're trying to give forestry managers to help them understand their risk. So as we move into Saskatchewan, maybe our southern pines in Saskatchewan aren't as at risk as our, you know, centrally located pines. You know, we get into Ontario, you can see there's a little bit of a patchwork. So maybe if there is the risk of spread, we start focusing on those stands with less resilience first for management, because there are some management actions that can be taken. And then I've primarily been talking about natural spread, but what about accidental introduction? So we have mountain pine beetle in the in the west. You know, there's a lot of transport of wood, both commercially and personally. And while there are lots of policies in place, we all know that these introductions do accidentally happen. And what happens if that accidental introduction happens in Ontario is we actually have additional pine species that are going to be at risk. So in Ontario, we have jack pine, we have red pine, and we have eastern white pine. And Rosenberger showed that those species, eastern white pine and red pine, are both susceptible to mountain pine beetle. And so this puts our kind of jack pine in the north of Ontario and then our southern pine species at risk as well with accidental introduction. And so this is something that I'm going to be exploring a bit more in the next few years, using information from lodgepole and jack pine and seeing how it translates to these other pine species. And what are the implications for mountain pine beetle spreading? So if we think about managing invasive species, it's very expensive. So the ISC report from 2019, it showed that Ontario spends about 30 million annually on emerald ash borer. And a recent presentation by Erica Samus in Alberta showed that the government so far has spent 568 million to manage mountain pine beetle. And you can see in this graph on the bottom, the area disturbed in millions of hectares by mountain pine beetle over the past 10 years. When it outbreaks under these, you know, climate changing conditions, it is extensive. And so a province is looking at costs of managing it, but those costs aren't also including the loss in timber and the loss in forestry products. So there's financial implications in terms of management, but also financial implications in terms of the loss of these important species. And so what are the overall take homes? First off, jack pine is susceptible to mountain pine beetle as well as red pine and eastern white pine. So all of our pine species in Ontario are at risk. The rate of natural spread is still highly uncertain. It requires additional research to understand how mountain pine beetle is going to behave in the novel habitat. And that's where, you know, work like mine and others comes into play. The risk of accidental introduction of mountain pine beetle brings additional uncertainty to the system. You know, if mountain pine beetle arrive tomorrow in Ontario, are we prepared to manage it? And then finally, the potential cost of that management and the loss of timber to Ontario would be extremely significant. You know, jack pine's the third most abundant tree species in Ontario and is an important part of our, you know, of our timber supply. So there's lots of implications for this, both from, you know, a forestry and economic perspective, but also from, you know, ecological and environmental perspectives. And with that, I'll thank all of the groups that have been entering into this project over the years and all the supporters of this research.
Thank you, Catherine. That was great. It seems like there's lots of things to consider when trying to figure out where mountain pine beetles can end up next. So thanks for sharing. I'm just going to switch over my screen. All right, so our next presentation is on traditional ecological knowledge in forest management, and I am pleased to introduce Claudette Trudeau and Travis Jones. Claudette Trudeau is an Anishinaabe, Ojibwe, Mukwudodom Bear Clan, a member of the Serpent River First Nation, and a second-generation Indian residential school survivor. Claudette earned a Bachelor of Arts in Sociology and has 30 years experience working in various federal departments in the federal government, including Employment Canada, Statistics Canada, and Indigenous Services Canada, specializing in Indigenous policy. She is currently with Natural Resources Canada as a policy advisor at the Great Lakes Forestry Centre. Claudette takes pride in supporting a more inclusive and collaborative approach on Indigenous perspectives in her policy work. Travis Jones is an Anishinaabe, Ojibwe, Mukwudodom Bear Clan, a member of the Garden River First Nation and a father of 3. Travis had a traditional Anishinaabe education that continues today. Travis has also earned a Bachelor of Science in Forest Ecology, as well as technical diplomas in forestry and fish and wildlife. Travis has extensive experience in both forestry and working with Indigenous people. He has worked for more than 10 years in forest ecology research at the Northern Forestry Centre, as well as another 10 years as Indian and Northern Affairs Canada. He is now the Indigenous Engagement and Science Advisor at the Great Lakes Forestry Centre. Travis has always been passionate about addressing the underrepresentation of Indigenous people in the sciences. Travis feels fortunate to work with Indigenous people through a natural resource development lens. And he leaves me with a quote. So, as an Anishinaabe Ojibwe man who grew up on the land surrounded by the many miracles of our Creator, it's an honor to work with so many brilliant and inquisitive minds here at Natural Resources Canada who share in the same sentiment of adoration that comes when we are embraced by the arms of our Mother Nature. So I want to thank you both, Claudette and Travis, for joining us this morning, and I'm very much looking forward to your presentation.
Great, I'm just going to share my screen.
Yeah, you should be good to go.
Are you able to see my screen now?
Yes, I see your desktop screen.
Oh. Okay, need to change screens. Monitor 2. Is this correct now? Can you see my screen?
There you go. It looks good.
Great, wonderful. Bonjour, welcome everyone. I'm so happy to be here to join you here for this great forum, especially, as Elizabeth mentioned, here on traditional territory, my home territory, the Ojibwe land here in Boating, Sault Ste. Marie. And as well, it's wonderful to have my colleague Travis and I talk with you and reflect on some of the work that we've done working with forestry and traditional knowledge. It was great to listen already to our colleagues and presenters. I know you had a great keynote speaker yesterday with Sue Chiblo, and as well our colleague David Young last night, who all have been talking about the great work that happens at Great Lake Forestry Centre. We also want to really emphasize, as Dr. Chris Macquarrie mentioned, we work closely with the centers across NRCan, but today we're going to reflect on some of the efforts that we have done here in Sault Ste. Marie. Kind of the purpose of our talk is we're going to reflect on the work, as I mentioned, and some of the knowledge that we learned working with some of the neighboring Indigenous communities, as well as identify and share with you some of the lessons learned that could be transferred to invasive species prevention and management and work in Ontario and beyond. Especially, as I mentioned, we do work closely with all of our centers across Canada and share all our experiences when working with the Indigenous communities. It'd be great today to really let you know a big part of the effort was our collaboration with the Invasive Species Centre. Around October 2018, we began our traditional and cultural knowledge experiences. We joined with local Indigenous communities, brought together some of the federal civil servants, as well as local Indigenous species of risk and communities across the province who all work in Sault Ste. Marie to really get together and creatively learn some hands-on experience. We were fortunate to start with the Kairos blanket exercise, and it continued on to some great work, especially with visiting here locally. We have the Shingwak University, a local residential school that is converted into now a learning center. One of the Truth and Reconciliation Commission's huge calls to action is for to have the number 57, where we're really looking at having all of our Indigenous issues brought to full attention and learning about Indigenous rights, Aboriginal law, and getting that competency up in a lot of our staff that are working. So with all our plans, we took over the learning of the 2 years was really based on training and learning some of the residential issues and reaching out towards the United Declaration on Indigenous People. I can't explain enough about how much learning happened with many of the activities that we had, exploring Indigenous culture, getting together with featured leader Dr. David Pearson, who works at Laurentian University, and bringing us together to do some thought on ways of learning, ways of knowing, creatively thinking of how scientists can work together with Indigenous and traditional knowledge. So much learning happened, especially when we involved our youth. A huge step was the internship program, which we brought to Great Lakes Forestry Center. And we were fortunate to have 4 interns last year complete a successful forestry ecosystem research and assessment team experience. A highlight of their internship included the presentation at Algoma University. The research in each of their experiences has flourished. As one of our colleagues has recently joined, and you had David Young's presentation yesterday. We're very happy when we learn the importance of including Indigenous youth in all of the aspects. I'm going to hand it over to my colleague Travis now. He's going to talk about some of the proper relationships we developed.
Hi, thanks, Claudette. Can you hear me okay?
Yes, it's coming in clear.
Awesome. Okay, so miigwech for asking us to present. It seems to me I do a lot of Indigenous engagement work, and I kind of, my conversation at work are drifting away from science, so it was the science nerd in me is enjoying the presentations today. Like Claudette alluded to, we're just, want to sort of tell the story of what we've done here at Great Lakes Forestry Center in terms of Indigenous engagement, and obviously invasive species is a part of that discussion. What I think lays the backdrop for our collaboration that we've had, it really embracing the Truth and Reconciliation recommendation, where it states that public servants should be trained in Indigenous culture and history, just so Canadians and more importantly, in this discussion, public servants and others, and I would say the science community, just recognize the uniqueness, the unique relationship between Canada and its Indigenous people, and for a science discussion, how Indigenous people see the world and how our more Indigenous approach meshes or works with a more Western science approach. So it's important to remember we have we being in the Indigenous community, have obviously a strong connection to the land and are very familiar with our surroundings. Our Indigenous knowledge is based on, many generations of observation. It tends to take a more holistic approach, but there's some things that we don't know a whole lot about, and invasive species is always one that comes up. In our language, Claudette and I are both Ojibwe. In our language, there's no word for sea lamprey or emerald ash borer or smelts or rainbow trout or things like that, because it's something that we haven't seen. So our Indigenous knowledge lacks in that, obviously lacks in that invasive species conversation. It's important, I think. So next slide. Well, I don't know if we went. Okay, I think it's important to just have a quick discussion here about how that quote from Leroy Littlebear, I think, is another way of saying sort of what the TRC said about if we're going to work better together, then we have to sort of recognize how our culture is different from a more Eurocentric culture. And just very quickly, we'll get through this really quick. Land is sacred. It's a gift from the Creator, as opposed to being sort of a resource or a commodity. There's a good example of this in our community, of my community of Garden River, where our community was very reluctant to do the sea lamprey, lampricide application on our river. Our community sees our river obviously as a living thing, as a person, for lack of a better term. In the Ojibwe language, things are divided sort of in our language, much like French has a masculine, feminine. Well, to Ojibwe people, we have sort of a living, non-living, and it's really interesting how we see things. So rocks aren't living, but trees are. So the way we talk about it, by our pronouns and stuff, whether or not it's living or not. When you talk about water, we talk about water like it is alive. So with this approach, if water's seen, and particularly water bodies like rivers, seen like having its own spirit and being a relative of ours, then how can we put poison into a relative of ours? And I think through collaboration that we were a part of, I think that the community sort of started to see more learning about the invasive species and the detrimental effect of something like sea lamprey. And really, I think this is the best tool, the greatest tool we have against this invasive species. So for the first time last year, the community did allow lampricide to be applied to our river, which to me signaled a greater relationship. We did a lot of work with Garden River that you hear about coming up in terms of addressing some of the distrust that exists in Indigenous communities, and now they see what was once seen as poison, now they see it more as medicine. And I talk lots to both my Indigenous community and my science community. As my wife had thyroid cancer, it's behind her, everything's good there, but the stuff she had to take, that chemotherapy, was so nasty and so toxic that it was very, very scary for our family to go through. Watch my wife ingest this stuff, but in the end, it made her better. It was medicine, right? So it seems like the community is sort of starting to embrace the science community a little better, and seeing things like what was once seen as poison, maybe more now is seen as medicine. Just an important part here, the world is seen as interconnected and holistic, that can't be dissected, clashes a lot with this compartmentalized sort of Western approach to things. Next slide, Claudette. So one of the things we did, I think that we do, again, just quickly, when we started engaging more meaningfully with Garden River First Nation and others, they felt that the discussion was on the government terms, on the outsiders' terms. You come to our building, we'll sit in a sterile government boardroom, and we'll talk about how we can work together. And the community said, really, Travis and Claudette, if you want to have a discussion with our people and our knowledge keepers, you should have that next to our river, underneath our white pine stand, at our pile grounds. So that's exactly what we did. We sort of went out of the box and arranged a tour of some of the sites that Garden River that meant a lot to that community, and really had a great discussion about. Sorry, I'm getting a lot of feedback. Had a great discussion about the what meant a lot to the community, and the community felt like, well, they listened to us. They want to be better partners with us. They listened to what we had to say, and it really felt like some of that distrust was less of an issue. And going forward, it really felt like true collaboration. We were on something that was a path of true collaboration. Next slide, Claudette. So this is just a picture of one of the lands we had on the Garden River. This is at the 10 trailer park under the old growth stand. And that picture it was important for the community to include the youth and teachers that day. So it wasn't just elders and knowledge keepers. It was a good cross section of the community mixed with a lot of scientists from the Great Lakes Forestry Center. So I think that picture sort of symbolizes, a good way forward. There's no nobody at the head of the table. There's nobody above or below anybody. That day was really special, and there's a lot of a good, meaningful collaboration that's going to come out of doing things like that. And I think the success of days like that was we listened to the community. We heard what their priorities were, inadvertently that addressed some of the mistrust and sort of some of the legacy issues that comes along when outsiders or government engages with Indigenous communities. And I'm really excited about the future. Invasive species obviously is a big concern for Indigenous communities, particularly at this community, because there's salmon and smelts and emerald ash borer and sea lamprey. Some of the things that come out of that day from an invasive species lens, I think that was interesting. It's hard for Indigenous communities like ours to understand why we manage invasive species or promote them with salmon and things that we fish for, but other things we don't like sea lamprey and emerald ash borer. Whereas to us, we are more holistic approach to things. The sort of Western approach to the management or approach to those visitors sometimes clashes with the Indigenous approach of you could cut a tree without the forest knowing, I think, is the central dogma to our approach to development. So to manage one species but then eradicate or try and kill off another is something that represents sort of a hill in front of the science community when engaging with Indigenous communities. And I think it's exciting going forward. Another thing that dominated or that was a part of that day is, for the longest time, we were the Indigenous community felt like they were treated as a stakeholder, whereas Indigenous communities aren't a stakeholder; they're a rights holder. And there's a great distinction between those two. So as the government and outsiders do things that could affect the rights of Indigenous people, well, we're a necessary part of that conversation. The courts of Canada certainly have supported that notion in the last decade or so. So I think going forward, this conversation between invasive species and approaches and management of invasive species and Indigenous communities is going to get more complex over time. And I think some of the approaches we've taken here at GLFC certainly cuts a nice trail, an exciting trail that we could follow to have greater collaboration and in the end have a stronger partnership that benefits everybody. If I didn't, Claudette, did I miss anything? Anything to add? Oh, you're on mute, I think.
And, oh, sorry. Yeah, we definitely want to work closely with the Invasive Species Center, and stay tuned. There will be a compendium of activities that have been learned from lessons across the country as we are learning at NRCan on really some great relationship building that has happened across the country on working with Indigenous people, as well as in the sciences. As Travis mentioned, listening is the biggest lesson that everyone is learning from this. So important to take the time and build that relationship. I left for you the contacts to reach Travis or I want to say thank you, miigwech merci for today, and looking forward to any questions anybody has at the end of the presentation. Thank you, miigwech. Bamapi.
Miigwech, thank you, Claudette and Travis. That was great. I think, collaboration into the future is going to be very important, to deal with, climate change and invasive species and all the environmental impacts that are here and could potentially happen in the future. So thank you. So our next and final presenter during this session is from Dan Rowlinson. And he will be presenting on forest health conditions for Ontario 2020. Dan Rowlinson is the provincial lead for the Ontario Forest Health Monitoring Program, beginning his career in the mid-80s as part of a spruce budworm research team. He now has the role of planning the provincial level forest health monitoring program with the Ministry of Natural Resources and Forestry. Currently living in the Algoma region, which is also home to Natural Resources Canada and the Invasive Species Center, he continues to nurture collaborative partnerships with the focus on monitoring and research of the growing list of threatening invasive species. Forest health monitoring in Ontario is conducted by the Ontario Ministry of Natural Resources and Forestry. The forest health monitoring in Ontario includes the occurrence of native, non-native, and invasive biotic and abiotic disturbances and events. All forested areas in the province, regardless of ownership, is monitored and reported on each year. So thank you, Dan, for joining us this morning, and I'm going to hand over this for you to share your screen.
How is that, Kristen?
It looks great.
Okay, thanks very much for that introduction. And so since we've started this the forum earlier in the week, we've heard a lot about invasive species, and for the layman coming in would sort of look at that and say, we are doomed, man. We don't have a chance of surviving. Our trees are not going to survive. It's really encouraging to listen to the talks, and you know, it gives me great comfort in knowing the bench strength that we have in Canada to deal with these invasive species. And we've heard a lot of key words. I think, you know, as we talked about in the opening session with Gary Pritchard mentioned the importance of sharing with other generations, and he referred to them as those fireside chats. And I think that, you know, the time has never been more important to do those conversations and carry those conversations and pass this information, both the scientific and the information on our native pests onto the next generations who will have to deal with this, as we hopefully are able to leave them with a fairly sustainable resource as we move on. So thank you very much for that. So what I'm going to talk about is the native forest pests in Ontario, and we talked a little bit about the Forest Health Monitoring Program, and I do work closely with a lot of the researchers that I've presented already in the data we collect. Our program is it's a provincial level program. It has a regional delivery model, so we have staff in the Northwest region, Northeast region, and the Southern region, who all collect information that feed into the much larger provincial scale, and we produce a report at the end of the year, basically what's going on in the landscape of Ontario. So some of these insects that I talk about are native pests, and you know, one of the key words that we talked about earlier in the week, and Minister Yakubowski opened with, is the importance of biodiversity and protecting that biodiversity on the landscape. And some people, you know, and even sometimes myself, you know, we need to be reminded that a healthy, vigorously growing landscape includes outbreak cycles of insects. It includes mortality to trees. It includes these large forest fires that we have in the northern areas. In order to maintain that biodiversity, refresh the landscape, and provide, you know, other species opportunities to start that living process over again. Forest ant caterpillar is a really good example of one of our native pests, a cyclic insect. 2019, we had about 35,000 hectares of defoliation, and I'm really happy to mention that in 2020 there was no mappable defoliation during our exercises or our surveys. So our data that links back to the early 50s, you know, you can almost set your calendar on these 10-year outbreak cycles. So we're in the downswing of the forest ant caterpillar outbreak in the province right now. Spruce budworm, it's another native insect. So it's a pest that has these native cycles on the landscape. It has, you know, our data again dates back to the 50s. It was you know, the subject of many large-scale operational aerial spray programs in the 70s into the 80s. It took more of a management or a localized management response moving into the 90s and 2000s. This gives you an idea of one thing I like to mention in this chart that you're looking at right now is that we've come a long way in our data collection techniques that we have in the field. So these areas that are up in the 20 million hectares of defoliated forest stands, you know, in using today's methodologies with the digital mapping tablets that we have as compared to the sketch mapping that we've done historically, I mean, it was the best product of the time, but we have much more refined data collection techniques. So that's why there's such that such a range in the landscape level disturbance mapping that we have. 2019 in the Northeast region, was where the bulk of the spruce budworm defoliation was, about 340,000 hectares of defoliation. That area expanded considerably into 2020 with about 440,000. It's become the subject of a planning process in the Northeast region, potential operational spray programs, some accelerated harvest in some areas, and there will be a large area of this forested stand will be left to run its course from the budworm outbreak cycle. Another native forest pest that we deal with on a regular basis is jack pine budworm, particularly in the northwest part of the province. This is a pest of the jack pine on the landscape. It's an insect that in 2019 caused about a million hectares of defoliation. This one's fairly significant. We know how the economic value of jack pine on the landscape is to our forest industry in the north. And as we know, we all need 2x4s to build our houses and provide that lumber opportunities. And the economic value that's attached, as well as the biodiversity value of this jack pine species, is very important on the landscape. Outbreak cycles, again, the data we have on jack pine budworm dates back into the 50s as well, and we've had large spikes in the 80s of the jack pine outbreak, more smaller peaks and valleys moving into the 2000s, and we're just coming through an outbreak cycle now. 2019, there was an aerial spray program conducted in the northwest part of the province of just over 100,000 hectares was sprayed with BTK. Not to eliminate the insect, but to provide the foliage protection to the jack pine trees throughout the natural outbreak cycle while it collapses. Again, in 2021, there is a planned exercise to conduct an aerial spray program on a much smaller scale. About 75,000 hectares of forest is scheduled for control in the northwest part of the province. Gives you an idea of, and this line up here is not a bug fence, so I'll just bring that up right now. That's basically below this area is the area that we concentrate on because it is the managed forest in Ontario. So we, when we're budgeting our survey budgets, we focus on the managed forest, and this is an area that we had jack pine budworm in 2019. As I mentioned, that million hectares, this is where it was concentrated in that time frame. This is a little closer scale of the outbreak. This is in 2020. We know we're sitting now at about 930,000 hectares. When you're going through an insect outbreak cycle, the way the outbreak looks on the landscape is just as important when it's building as when it's on the waning years. So when you start to see the populations breaking up, these smaller satellite areas of defoliation and not so severe on the boundaries, that's an indication that potentially you're starting to see some collapse of the outbreak cycle, and that's sort of where that graph that I showed you provides those peaks and valleys. And jack pine budworm, I would expect in the next few seasons should be basically on the downswing in Ontario, and we should be free from jack pine budworm moving forward. One thing that we do to support control programs and also to verify our data that we're collecting in the field is this is a jack pine budworm example. It's a second instar larva survey where we go out in the wintertime and collect branch samples. But describing my perspective that I showed you that last map, you can see all these green areas that although they were defoliated this season, this is a predictive forecast for 2021. The area of defoliation should be significantly reduced based on our forecasting information. So we use these tools we have to predict some of the pests that we do have on the landscape. This gives you an idea of when we go out in the landscape in the wintertime, collect our branch samples from the upper mid portion, bring them back to the lab here in Sault Ste. Marie for processing. We soak them in a solution of sodium hydroxide, and the budworms, when they overwinter, they crawl underneath as a tiny second instar larva under the bark scales on the branches, and they spin themselves a nice winter hibernacula, it's called, or a wool sweater. When we run them through this process, this enables us to burn that hibernacula off them, and we're able to count them under a microscope, plug that information into a formula, and it will predict us what the defoliation will be for the upcoming season. It just enables us to provide a more focused approach to a management response. Gypsy moth. Our Southern Ontario folks will know all about gypsy moth. This is an invasive species. It's an important invasive species because it's one that we look at. We have data in Ontario dating back to the 60s, as I'll show you the graph, and it's one that is seemingly naturalized. It's still an invasive species, still under a quarantine restriction with the Canadian Food Inspection Agency, so it is a regulated pest, but it is one that according to our data and our field observations, has basically naturalized. And when I say naturalized, it's been in Ontario long enough that those control agents that are present in the population, the funguses, the diseases, the parasites that cause a collapse in a population have evolved in this population we have as well. This gives you an idea, and you'll see this summer where the defoliation peaked at almost 600,000 hectares. So our data in Ontario dating back to the 80s when it first started on the landscape, causing widespread defoliation, and then into the early 80s and 90s, and then it was in abeyance for a number of years. And then we've seen peaks and valleys. We haven't really seen anything like we saw this past summer for a number of seasons. 2019, the area was damage was concentrated mainly down in the Golden Horseshoe area, about 42,000 hectares of defoliated forest stands, woodlots, municipalities, and ornamental trees. 2020, remember when I talked about that breaking up of a defoliation back with the jack pine? If you look what's going on down in this part of the province, we're still seeing fairly high forecasts, but we're starting to see parasitized egg masses on the trees. We're starting to see dead larvae throughout the growing season that are infected with the virus and the fungus. So this area down in here is going on into 4 years defoliation. Based on our historical perspective, we would anticipate we're starting to see some collapse in the population down here. But as one of my mentors in my early forestry career told me, never make predictions, Dan, when it comes to insects. And that was Dr. Gordon House. And I hold that phrase in the back of my mind all the time when I'm talking to land-based managers and trying to predict what's going to happen in the coming season. So this gives you an idea in Ontario where the bulk of the defoliation is now. And if you see this area around Sudbury, this is up in the rocky outcrops, the hilltops. We had an area of defoliation quite similar in 2015, had extremely cold temperatures for a number of days. I think the span was about 5 days. And there was very little snow that year, quite similar to what we've seen in 2021, the winter of 2021. And you could not find a larva out there the following season. So it was very susceptible to the extreme winter that we had that season. So just some interesting factors that take into play when we're talking about these species on the landscape. Again, I mentioned it is a regulated pest, gypsy moth, and it is involved in a quarantine. This quarantine area, the northern boundary, has been extended this past winter, so it now includes the entire Algoma region. We talk about collaboration, and I know as all the discussions that we've talked about, there's not one agency that can tackle these invasive species threats on their own, and it requires these fireside chats, if you will, like we're having this week, to talk about what can we do, what are you doing, how can I help you, how can you use the data that I'm collecting in the field. We support some of the trapping programs that our federal counterparts of the Canadian Food Inspection Agency carry out outside of the known area range of gypsy moth. These are deploying traps basically from northwest to the Manitoba border from the known area, which is around the Wawa area. So when you're looking at the mapping these disturbances, Dan, you might ask, what are you seeing from the air? Are you, it's not like driving your car down the road on a summer evening and you stop at the end to get gas and you have to clean all those bugs off your windshield. That's not how we do our data collection, although that does provide a pretty good perspective on the aircraft. But when we're looking down at the woodlots and that, particularly in the southern areas, this is what we're seeing. So we're flying grid lines over these affected woodlots and these stands, and we're actually mapping this defoliation on our digital mapping tablet. Much more severe area, but you can see the difference in the transition. The gypsy moth around the mid-range part of your screen, defoliation of hardwoods, you get into a change in species on the hilltops there, and there's no defoliation. And then you get back down into the lower regions at the bottom of the slide where you're seeing more defoliation. So it's important when you're collecting this data from an aerial observation perspective that you take all those facets into consideration when you're determining the causal agent. Hilltops, it's always a telltale sign when you're flying across some of those areas around the hills of Bancroft, north of Peterborough, and the southwest in the Renfrew area. You see a defoliated hilltop and you're in a gypsy moth outbreak, you're pretty well sure it's going to be a gypsy moth that's causing that if forest tent caterpillar has collapsed. So this, again, these hilltops look like wintertime, but it's defoliation by insect. So a huge part of the work we do, and we're talking about these defoliations and the pending management responses that can be linked to your survey data, is we do go in and do a subsample of the data we collect, and we collect voucher specimens so that we know the damage level has been assessed on the ground, not just from aerial observations. And this insect, a life stage of this insect has been verified by our partners that carry out the insect identification for us with the Invasive Species Center, partnering with the Canadian Forest Service to provide that to our program area. So we collect the stand parameters, the host species, the life stage of the insect, and we provide a guess of what we think it is, and then it's verified by our partners in the lab. I talked a little bit about life stages. Gypsy moth is getting, it's an invasive species. It's really getting a lot of publicity this last year because it's been so intense, and it's an insect that is very disgusting. It's close to forest tent caterpillar, but when it comes to crawling all over you when you're sitting on your back deck having your coffee in the morning, but it's a much larger insect. Egg mass size is something we look at when we're predicting populations for the coming year. We see this female gypsy moth laying a nice egg mass there, but it's a small in size. As I mentioned, these inverted V's are telltale signs that these larvae, they've stopped feeding mid-season and they've been infected by a virus. And if one thing we have learned in Ontario, it's the impact a virus can have in a population and how quick it can spread. Whereas gypsy moth, when we're dealing with our people populations, we want to tell them, stay home, wear a mask, don't spread that virus around. When it comes to our gypsy moth friend, we want to make sure that they're climbing all over each other and, during their grooming period and their feeding periods, and they're singing and doing everything they can to spread this virus, because the more virus that gets spread in the population, as we know, will have a negative impact on that population and expedite that collapse. So I talked about the tool we have for forecasting in the budworm world. So what we do for gypsy moth is we go into these stands and we basically count the number of egg masses that's representative on a tenth of a hectare, plug that information into a formula, and that gives us a predictive population for the upcoming season based on egg mass density, numbers, and health of the egg masses. A very nice healthy egg mass you can see on the main stem of the tree. And when you're looking at these egg masses, it's also important that we calculate or tally where they're located. Remember that scenario I mentioned in Sudbury a couple of years ago where we had that huge outbreak? Egg masses that are below the snow line are going to have a better chance of survivorship than the egg masses that are above the snow line on the main stem of the trees, because they're in a normal winter, an average winter, they're protected by the snow load. This gives you an idea of our egg mass survey that we did this season. So we collected, did our Calidar plots, or these at a number of locations. Some of the forecasting that we've collected, the bulk of it is going to be severe, but as I mentioned, some of the parasitism levels that we had were focused down in this southwestern part of the province. Some of the other incidental pests that we did report on this season: large aspen tortrex. There was a small increase in the northeast part of the province to about 22,000 hectares, and also Manitoulin Sault Ste. Marie. We did have an outbreak of that. This gives you an idea of another defoliator. This is the large aspen tortrex, and as you can see, it's a messy defoliator. When I talk about messy defoliator, when you're flying your grid lines, you can look at the landscape and you can say, well, there's something going on with those trees. It's not totally denuded foliage like the gypsy moth does or the forest tent caterpillar does. It's more of a ragged, leaves bundled up, chewed a little bit. It's a messy feeder. So this is something that we would go in and verify on the ground just to confirm that that's in fact what it is. And in this instance, this is something we would sample and send in for identification. The moth is fairly significant because it can be confused with spruce budworm moths. If you look at the top right of the screen, you can see the adult spruce budworm moth. And in the main center of the screen, you can see the large aspen tortrex. Very similar, but again, that sort of stresses why when we're evaluating or assessing an insect disturbance, it's so important that we make sure we have the data verified. This gives you an idea of on the ground what a difference in defoliation as opposed to these whole tree total defoliators, a much ragged appearance that large aspen tortrex causes. So we had in one of our research presentations some of the work on white spotted sawyer beetle. So sometimes you can see white spotted sawyer beetle from the air. Reason I plugged this one into my talk today, you know, the area was quite small. It was about 4 hectares. It was close to an old burn. But this is one of the beasts that's commonly confused with the Asian longhorn beetle. And when we talk about our citizen scientists and how we find these invasive species, most times on the landscape is somebody found something that, you know, I saw this in a presentation or a magazine or on a sign outside of the city, and I think this is what we have. So white spotted sawyer beetle is an example of that. This is the damage that it can cause. You know, it's another longhorn beetle. It's a wood-boring insect, and the sawdust that's produced as it's crawling out of the tree and it's mining under the bark to lay its eggs. Oak decline. Dr. Sharon Reed talked about oak wilt. Some of the work that's been being done in Ontario regards to oak wilt. So when we get a call from the public or from one of our regional district offices saying, you know, we've got a report of some declining oak in this area, can you guys come and investigate it? So we take those investigations quite seriously because we know that that the best response opportunity to something like oak wilt is the early detection, providing that rapid response approach. So this was an area that we had down around Serpent River, about 570 hectares of oak decline, not oak wilt. And there were a number of factors at play in that stand. This gives you an idea of a root rot disease, armillaria root rot, that basically works along with Mother Nature when these trees are stressed or decline for you know, some reason of, you know, growing on a poor site, very dry conditions. Armillaria root rot comes in and plays an important key role of getting these trees back to the soil. One thing we've noticed this past season in the world of forest health, and you know, my small world that I talk about it basically runs from Quebec to Manitoba border and south, but the pictures that we get in and the questions to our MNRF science website, you know, we talk to all the as many people we can when these pictures come in. But I'm telling you, there's a lot of really good photographers out there that collect information, and you know, they send you these lovely pictures of these moth larvae. What is this? I found this on my my back deck. Can you help me it out? Is it going to eat my house? Sugar maple producers see these borers across some of their forest stands. What is this? Can you tell me? Like in the right there, where the gallery going across the tree and weakens the stem and prones it to blow down. The bottom left, something like white pine weevil. You know, why is the top of this jack pine tree dead? You know, and you look at the defoliation on the bottom right of something like a large case bear, which is another invasive species introduced into North America in the early 40s. You know, what's going on with these trees? They should not be red in May. So people were home a lot this season, and I think that's provided people with an excellent what I'll call reboot, enabling them to actually spend that time in the green spaces of Ontario and learning what's going on in their backyard and not having to run the gauntlet of a commute to work every morning in the dark, coming home in the dark. They've actually had time to have a look around and see what's going on around them. I'd like to draw your attention to the Ontario.ca site, where you'll find our Forest Health Conditions Report. 2019 is up there, and you can read on all the smaller insects. There's much more information that I've provided you today in this short period of time that can be found there. There's also the gypsy moth information from the survey we did, as well as the upgrading to the forecast work that's going on. And we're now working on fact sheets to supplement that Ontario.ca website. So on that note, thank you very much for your attention, and I'll pass the screen back, and thanks very much for the opportunity to present.
Thank you, Dan. That was great. Lots of forest pests out there that are impacting our forests. So thanks for that wonderful overview. I am now going to get us into our question period. So if there's a question that you have, please type it into the question box and I will do my best to ask it and get it answered before the end of our session this morning. So I'm just gonna pull it open. All right, so I have a question for Sharon. How can people report if they see signs and symptoms of beech leaf disease? Is this something that researchers are interested in?
Yeah. Right, so hopefully you can hear me. So currently, I have been directing people to Dan Rowlinson and his group so that they can go to the locations and collect samples and then have them sent into our lab. We're mostly focused on confirming beech leaf disease in areas where we have, like counties where we haven't detected it yet. We have a pretty good grasp on how widespread it is in the counties that we've already detected it in.
Okay, great, thank you. I now have a question for Catherine. Do you think a monoculture type forest is contributing to the spread of the beetle? And do you think forest managers can help with prevention by using a more diverse replanting adaptation?
Yeah, that's a great question. And definitely having a monoculture is not going to help. And the outbreak in BC was more like everything was managed to be the same age for forestry, right? Because they need to chop down the trees at a certain time and it's easier to have them all at the same age. So that was part of what contributed to the extent of the outbreak. So having, you know, uneven aged stands as well as the diversity of trees will help make your forest more resilient. So definitely having, you know, a mixed planting is ideal.
Okay, thank you. And for Dan, it looks like there was a severe infestation of gypsy moths in the area around Frontenac and Kingston. Can you confirm?
Yes, that area is included in the overall provincial forest defoliation level this season. It's moving into its second year defoliation in that area, so that's where the new areas has been recorded this season.
Okay, thank you. And for Claudette and Trudeau, I just have a comment just saying that they would love to learn from your experiences. And that's from Heather Charles. Okay, are researchers interested in tracking eastern hemlock across all of Ontario as an inventory for the potential spread of the adelgid? Not sure who wants to take that one on. Maybe Chris?
Yeah, we're certainly interested in knowing about where hemlock is. Kathleen Ryan with Silvacon has been accumulating existing inventories of hemlock that folks have sort of at the county level and at the like with the conservation authorities, and then we have a project with research at the University of Guelph looking at using remote sensing tools to develop a comprehensive inventory for Southern Ontario.
Okay, great. I'm not seeing any more questions, or maybe there's one. I do have a few questions of my own for some of you, so I'm just gonna ask a few. I'll start with you, Chris. So I was just wondering if you could talk about the process for finding biological controls and, you know, the approval process and how long this can take.
So the process of finding them usually involves going to the native range of the insect. So in the case of emerald ash borer, that was parts of China, and in the case of hemlock woolly adelgid, it's also different parts of Asia and also Western North America. Hemlock woolly adelgid is native to Western North America. So going to those places and looking for those things that eat the insect, and then the next step is once you have sort of what you think are candidate species, you bring them back to a lab like we have at the Great Lakes Forestry Center and bring them into a quarantine situation so they can't escape, and then you perform various kinds of host. You assess their host range, so you're looking to see how do they just eat the insect that you're interested in targeting, or do they eat a bunch of other stuff, and how well do they eat those insects or parasitize them. And then on the basis of that, you can assess the risk of introducing that potential biological control agent to a new place. And then in Canada, once you have that information, you write a petition to the Canadian Food Inspection Agency for permission to bring that insect into Canada and then release it into Canadian ecosystems. And then so then that petition goes to what's called the Biological Control Review Committee, and that's a committee that's of scientists and experts who are knowledgeable of all of biological control and insect population dynamics and taxonomy and various other professionals. And they review that petition on the basis of how good the data and how well have you assessed the risk, and then they give advice to the Canadian Food Inspection Agency as to whether or not the evidence is scientifically sound. And then the Canadian Food Inspection Agency takes that information and takes that assessment and then does their own process looking at other values of risk and other things that are important to them, and then makes a decision as to whether or not it's safe to release that into Canada. And then after you have that permission, then the next step is trying to figure out whether can you rear it in big enough numbers, can you release it into the environment. So that, just by me describing it, obviously sounds like it takes a really long time, and it does. Between the time emerald ash borer was found and the time the first parasitoids released was about like 6 or 7 years in the US, and it was in 2012, so it took 10 years to get permission to do the first releases in Canada. It's for hemlock woolly delgid, it's probably about a similar length process.
Okay, thank you. That was great. I'm just going to ask one more question as I see it's coming to the end of our time here. And I have it for Sharon. And I was just wondering, I found it very interesting with beech leaf disease that a nematode was involved in transferring the disease. And I was just wondering, are there any other diseases that involve nematodes in transferring diseases and impacting our forests?
I always have to remember to unmute myself. In North America, there are agricultural diseases that are foliar diseases with nematodes. So this is one of the reasons it took us a while to understand beech leaf disease. Most diseases in forests in North America, that would be fungal in nature, and then if it's not fungal, then we start looking at bacteria and viruses. So nematode was last on the list, and we're still investigating if the nematode may actually be transmitting other disease agents as well. That's a possibility we still haven't ruled out, although just based on the evidence so far, it seems like that it might be slim. It may be more likely that there are some insect and possibly avian vectors that are helping these nematodes move around.
Okay, thank you. I'm not seeing any more questions coming in, so I think I'm going to end it here for today. I just want to mention that I just want to mention or thank all of our speakers for presenting today and for everyone for tuning in. Please join our final session from 1:30 to 2:30 this afternoon, where we'll be discussing final thoughts, next steps, and key takeaways from this forum. So I really hope to see everyone there, and I hope everyone has a great afternoon, and thanks for joining.
Thank you.
Thank you.
Thanks.
Thank you.